We're starting the molecular biology unit in this course. First, we're going to talk about some of the groundbreaking experiments that show DNA is the genetic material. Now, it seems obvious to us because we already know, but there was a time when scientists didn't really know the nature of genetic material. And there was within the scientific community a debate whether or not the genetic material is made up of DNA or it's made up of protein. They were pretty sure that the other biological molecules, like lipids and carbohydrates, were not making the genetic material was not made from those. But the debate was between DNA and protein, and the um, opinion of the scientific community was more leaning towards protein because if you remember, DNA is only made of four different types of um, nucleotides, but protein, the building blocks of protein, are 20 different amino acids. So the logical thinking was that since proteins provide more opportunities for diversity, and because there is so much genetic diversity, the genetic material would have to be made of protein. But then I'm going to show you some of the groundbreaking experiments that actually show that it is the DNA that makes the genetic material. And this also shows that just because something makes sense logically doesn't mean that it is the truth. So let's dig in. The first person that really showed the first experiments to indicate that DNA is the genetic material was Frederick Griffith in 1928. And his results weren't really taken seriously by the scientific community. And also, when he published his own data, he didn't really express so much 100% confidence in his own conclusions, unfortunately, but he really was the first one to show that the DNA is the genetic material. So, what did he use? He worked with bacteria that was used to infect mice, and there were two different types of bacteria. There were pathogenic bacteria that, when injected into mice, it would kill the mice, and non pathogenic bacteria which is closely related to the pathogenic bacterial cells, but when they were injected into mouse, the mouse would live. So there is something in this pathogenic bacteria that causes the death of the mice. So then he asked, what happens if I heat kill these pathogenic bacteria? Will they continue to be pathogenic? And when he heat killed it, so this purple one shows it's a representation of these pathogenic bacteria that are dead. But when he heat killed them, the mice lived when they were injected with it. Then he asked, well, what happens if I kill the pathogenic bacteria but mix it with the non pathogenic bacteria? Is there some factor in this pathogenic bacteria that can be transferred to the non pathogenic bacteria and then causes the bacterial cells the ability, the non-pathogenic bacteria, the ability to kill the mice, and that's what happened. So he heat killed the bacteria, pathogenic ones, mixed it with the non-pathogenic ones, and then inject that into mice, and he found that the mice died. So there was something in the pathogenic bacteria, some factor that was able to reprogram the non pathogenic bacteria to become deadly. So then he asked, okay, in these mice that are dead, there are bacterial colonies that are growing. So then he said, what if I take blood from these mice in which the bacteria are living, and if I grow the mice, is it going to be looking like the non pathogenic ones or looking like the pathogenic ones? And when he looked at it, it says it's as if it was it was an event where there's something in the pathogenic bacteria that just changed the nature of the non-pathogenic ones and gave gave rise to the growth of pathogenic ones. It was as if these bacteria transformed the non-pathogenic ones into what they were. So here's a results that suggested that there is a factor that you can take from one organism then and expose another organism to it, and it would reprogram that second organism, which is the basically concept of inheriting something. So then he did the experiments when he took these heat-killed bacteria, and then he separated the DNA portion from the protein portion. He showed that it was only when you take the nucleic acid portion, the DNA portion from the pathogenic bacteria, it is only then that you can get this result. It is only then that you can reprogram the non-pathogenic ones. When he took the proteinaceous part, the part that was made of protein, that didn't happen. So it was the first evidence that genetic material is made of DNA. But then the groundbreaking experiment that was really, really clear-cut and left no reason to doubt that DNA is the genetic material was done by Martha Chase and Alfred Hershey in 1953, which helped them win a Nobel Prize. And the advantage of the way they did it was the simplicity of the system that they used. They used something called a bacteriophage, and bacteriophage is an equivalent of virus but for bacteria. So these bacteriophages really do not have their own metabolism, and they rely on a bacterial cells to make more of themselves. And this particular bacteriophage that they used was really, really simple. They're only made of two different things. They're made of DNA and protein. The protein forms the outer covering portion, the body of the phage, and the genetic material was inside. It's inside the head. So the outside, if you like, the container is made of protein, which is holding the DNA inside of it. So how does a bacteriophage work? What is the life cycle of a bacteria? And we're going to start here. So the bacteriophage infects a bacterial cell. Now, at the time where these experiments were done, it wasn't really clear which part of this bacteriophage is reprogramming the bacteria. Is it the DNA part or the protein part? Of course, we know right now that it is DNA. So then what the bacteriophage does, it basically reprograms this genetic material of the phage, incorporates itself into the DNA of the bacterium, this is bacterial chromosome, and basically reprograms the bacteria to become a phage factory. It makes more of themselves, it assembles its proteins, it puts them together, and it makes millions of different phage, which ultimately burst the bacterial cell. Here's an electron micrograph. This is a bacterial cell, and these alien-looking structures are the bacteriophage that are infecting the bacteria. So this is the system that they used, and let's find out how they were able to do the experiment. But before we do that, we need to review something, and that is radioactive isotopes. So when we covered a little bit of chemistry, we talked about isotopes. So atoms of the same element can come in different forms, which we call isotopes. And these isotopes tend to have different number of neutrons. So all the different isotopes of an element have the same number of protons and the same number of electrons. But the number of neutrons is different. Now, since there is the same number of protons and same number of electrons, the chemical properties of different isotopes are exactly the same. So if you would put them into a chemical reaction, or if you feed it to an organism, the chemical reactions in living organisms can't really distinguish between the different isotopes because they are chemically exactly the same. Now, also remember that we said for every element, one isotope is always the most abundant. And the reason is because the other isotopes tend to decay over time into the one that is more abundant. So in this case, deuterium and tritium, they would lose these extra neutrons, and then they would decay to form the most abundant form. And what happens for some of these isotopes is that when they decay into the most abundant form, they release energy. Now, this release of energy allows scientists to track what happens to radioactive isotopes. You can imagine these radioactive isotopes as having some sort of GPS. Just like Google can track our cars when we drive on highways, these radioactive isotopes, by the fact that they're releasing energy, they allow scientists to be able to track them and see where they go, what happens to them. So, in addition to using a simple system like this phage, what Hershey and Chase did was that they used radioactive isotopes of phosphorus and sulfur. So why these two? Now, if you remember from what we talked about, the structure of DNA is full of phosphate. There's, especially in the backbone of DNA is made of phosphate, sugar, phosphate, sugar. So it's abundant in DNA and not very abundant in proteins. So what they did, usually the most common form of phosphorus is P31, but they used the radioactive form of phosphorus, which has an extra neutron, which gives off energy. The other radioactive isotope that they used was S35, which is a radioactive isotope of the more common one, S32. Why sulfur? Well, because sulfur is absent in DNA. DNA does not have any sulfur, but it's quite abundant in proteins. So, if they took phage that was not radioactive, so this would be the phage, no radioactivity, and if you would take a cross section of its head, you would see this. So these two are pretty much the same. This one is the cross section. So if they took a phage, and also if they grew bacterial cultures either in the presence of sulfur, S35, or in presence of P32, and then infected those bacteria with these phage, the new phage that is made in that bacteria will either be radioactive on its coat, protein coat, or it will be radioactive on its DNA. So this is how they were able to either label the DNA of a phage or label the protein of the phage, and then ask, okay, when the phage infects bacteria, which of these two items are going to enter the bacteria and reprogram it, and which one stays out? So here it is. They have a culture of bacterial cells in presence of P32 and a culture of bacterial cells in presence of S35. They made phage to grow on these bacteria. So these bacteria that are growing in here are going to make phage that have radioactive DNA. The phage that is made in this case has radioactivity in the protein, but not in the DNA. Then they infected these radioactive phage and infected bacteria in the absence of radioactive material. So the only source of radioactivity is coming from the phage. So they allow the phage to infect the bacterium and reprogram the bacteria. Then they separated the viral particles that are left outside from whatever that was injected into the bacteria. And the instrument that they used was your regular, the blenders that you find in your kitchen counter. So one of the most profound discoveries in science was done using your regular old blender that you would find on your kitchen counter. So I hope after this you won't look at the blender same way again. Anyway, so when they put this mixture, in each case into the blender, they were able to separate part of the phage that did not enter the bacteria from the bacteria that has now taken in whatever it was injected to it. Now they had to see where is the radioactivity going. And in order to do that, they would have to somehow separate physically what's left outside from what is left inside. And they did that by centrifugation. You basically say centrifugation involves spinning something really, really fast. Now, since bacteria are much, much heavier than the phage body, the bacteria are going to settle to the bottom, and the body of the phage that did not enter bacteria were going to remain in the soup. Now, what they found was that the radioactivity ended up being in the pellet, what's called the pellet in the bottom, only if the DNA was radioactive. And the radioactivity ended up in the soup and not in the pellet only when protein, part of the phage, was radioactive. So this suggested that the part of the phage that enters the bacteria is the DNA part and not the protein. So that was the groundbreaking experiment that finally settles the score on whether or not the genetic material is DNA or protein.